Ramachandran plots can be a bit confusing, and don't hate me, but I don't think they're actually that useful, except outside of like purposes for structural biology, where you're trying to see if your model fits correctly. But even then, you don't need to know what corresponds to an alpha helix. You don't need to know which regions correspond to beta strands. You don't even need to know what the phi and the psi angles, like which one is which. What is important though is that the Ramachandran plot is going to allow us to see that there are certain places that you see specific angles for different secondary structures. There are specific angles that are taken in alpha helices, in beta strands, even in anti-parallel versus parallel beta sheets. And all of this is coming down to the fact that the peptide bond is resonance stabilized. You can't rotate around it. You can't rotate like within it, so you can only rotate at the places around it. And so you get these two places that you're able to rotate these phi and these psi angles. When you have a peptide, you have like a chain of planes, and then you have these angles where things can rotate around them. We plot these angles out on a Ramachandran plot, and what we see is that if we go and we plot the angles for various peptides for various proteins, the points are all going to cluster in specific places because those are the places where there's not going to be steric clashing and where hydrogen bonding opportunities are going to be maximized. We can further say, okay, well, what amino acids are we more likely to find in what kind of structure than another? But for all of this, we don't really need to know exact angles. But by seeing things on a Ramachandran plot, we're able to kind of get this visual, kind of like this real sense that, yeah, these things that we're talking about at the like atomic level, they're actually playing out if we go and look at actual structures. So let's take a look at Ramachandran plots, and I'm going to go in more detail than you guys need to know because people keep asking me. They say they want to know more about these Ramachandran plots. So here it is, Ramachandran plots in all their glory, and I'll show you some great tutorials where you can play around on some active sites and like Proteopedia and cool stuff like that. So let's look. What we're looking at is a fragment of a peptide, and at its center we have the amino acid alanine, which is now highlighted. You can see that there is part of the preceding amino acid on its left and part of the following amino acid on its right. In the dark gray, what we have are the alpha carbons. So this is the alpha carbon of the alanine, and then the alpha carbon of the preceding amino acid, and the alpha carbon of the next amino acid. Now, you'll see that alanine is connected to those amino acids through peptide bonds. Remember, as the dotted lines are showing us, these bonds have resonance stabilization. The electrons are delocalized between this carbon, this oxygen, and this nitrogen. And this is going to make it so that it has partial double bond character. And if it has partial double character, you can't rotate around it. And so these peptide bonds are going to make it so that things are going to be confined to a plane. So you're going to have a plane that is defined by 4 atoms, by the one C alpha, the carbonyl carbon, the nitrogen, and then the next C alpha. So this kind of like backbone, these 4 is going to define the backbone of that plane. And then you have 6 atoms in total that are confined to that plane because you also have this oxygen and you have this hydrogen here. So you end up with this plane, and when you have amino acids joined together, then you're going to get basically a chain of planes. There are only specific places that you're going to be able to rotate, and we call these the phi and the psi angle. The phi angle is right now highlighted in green, and it's between the nitrogen and the C alpha, and then the psi is between the C alpha and the next carbon. I like to remember this as the psi kind of looks like a C, and you're going between the C and the C. So you're starting with a C. I'm going to a C rather than an N and a C. But basically, there are these 2 places that you can rotate, and that's all I care that you know. I don't care that you know the names, which is phi and which is psi, but I do think it's important to know that there's only a couple places that you're able to rotate. And it's important to know this because, well, that's going to make it so that certain backbone angles are preferred, and thus certain secondary structures are preferred.
When we talk about these angles, we call them dihedral angles. Dihedral angle is just an angle between two planes. And so as we see, we have these two planes and we have angles that we're going to be rotating around. So we're going to call these dihedral angles. Okay, so we have alanine. It's connected through these peptide bonds, gives us these planes. And now let's see what happens when we rotate them. In order to best see what's going to happen, we need to basically show things a little more like what they actually are. Right now, we're just showing these sticks, and so it looks like there's plenty of room to rotate, right? But what happens if we show the van der Waals? So basically, this is showing us how much space these atoms really are taking up. So remember that it's not just like a little proton. You'll have the proton surrounded by these big electron clouds. And basically, the van der Waals surface is going to show us kind of like the atomic radii. And what's going to happen is we'll see that as we turn the peptides at those phi and psi angles, what's going to happen is you're going to get clashes. Basically, we can't have overlapping of the electron clouds of atoms that are not covalently bonded. And so when we do, we call this a clash. We can see that as we change the angles, what's going to happen is there's going to be some places where there's bad clashes. So we can see that pretty much all these are going to be really, really bad. And there's only really going to be a couple places where things are going to actually not clash. So remember that clashes is physically impossible. You can't have those electron clouds be overlapping of those non-covalent atoms. And so what's going to happen is now there's only these few places where you don't see those clashes. And this corresponds to the beta strand region. It's really nicely illustrated in this tutorial. And so I recommend that you check out this phi and psi angles tutorial, and well, I actually require that you do that for my classes. But thank you, Eric Martz, for putting this together. It really nicely illustrates things. And so basically what we were just looking at, those angles where we didn't see those clashes, that corresponds to what we to the beta strand region. And what we're seeing here is a Ramachandran plot. A Ramachandran plot, basically what it does is it plots those phi and the psi angles. And if you do this for like protein structures that have been solved, you go and you look at all the atoms and you look at all their angles, and then you were to plot them on a chart like this, what you'll see is that the dots are going to cluster. Those dots are going to cluster in the regions where those angles are preferred, because only in those preferred angles are you actually able to exist. And so you'll see that there's actually, though, it's not just like there's one angle that can exist. There's kind of a range of angles that can exist. So why do you get this clustering? Well, you get this clustering in part because you also have to think about the fact that the secondary structure, in order to form strong secondary structure, in order to form those alpha helices and those beta strands, things don't just have to care about how the backbone angles are in order to avoid those steric strain, those steric clashes. They also have to think about how they can optimize their hydrogen bonding. So remember that hydrogen bonding, you have to be aligned to have it be optimal. Well, there's only certain arrangements that are going to, certain angles that are going to be more optimal for allowing the best possible hydrogen bonding in those strands and in those helices. And so this is why you kind of get things confined to smaller regions of the Ramachandran plot. And the angles themselves are also going to be restricted by the size of the R group. So with alanine, it's one of the smallest other than glycine. It's the smallest other than glycine. And so we'll talk in a minute about how glycine's a weirdo, but if you had things that were bigger, well, now they would have confined angles even more. But you can really see these core regions where amino acids are typically going to cluster. So this is a really great kind of interactive demo that you can do if you want to look at this. Basically, what it's showing you is the phi and the psi angles. So just like we saw on our Ramachandran plot, except here it's showing it for like a smaller molecule. So just instead of taking it from like 100,000 high quality data points like we were looking at in our chart before, this is taking it for a structural model. We can do this for basically any model, and I'll show you how you can do it if you really want to. But right now, let's just look at the simple thing. So this protopedia is a really great resource if you're interested in kind of like exploring proteins and exploring protein structure. Right now, what you're seeing is a structural model of an alpha helix. So if you click on these things in the article, it's going to kind of change the picture that you're going to see over here to highlight different things. For example, now you're going to be seeing those peptide, those planes of the peptide that are going to be highlighted. And what we can do also is we can actually plot the Ramachandran plot for this. We'll see that the points are going to cluster in one region. And this is the region where you're typically going to find alpha helices, the amino acids for alpha helices. And I don't think it's very important to know the actual angles that you're going to find, or even to be able to look at a Ramachandran plot and be like, oh yeah, that's alpha helix region. But I do think it's important to realize that, well, in an alpha helix, you're going to have these same angles, and this is why they're all going to kind of cluster in a certain region. And so basically there's a region where the alpha helices cluster. And what about beta sheets? So here, when we click here, now we're going to see some beta strands. We can draw the planes on here as well, similarly to how we did with the alpha helix. We can also, if we want, we can zoom in. We can take a look at those hydrogen bonds. So remember, that's going to further refine what the phi and psi angles are. So we'll see slightly different phi and psi angles if we were to look at an anti-parallel versus a parallel beta sheet. But here you can see we get that nice hydrogen bonding, and we can go ahead and we can plot this on the Ramachandran plot, similarly to how we did for our alpha helices. But you'll see now that the points are going to be clustering in a different area of the chart. And all of the amino acids that are in those beta strands are going to kind of cluster in the same area. And so now if you imagine doing this for lots and lots and lots and lots of models, then what you see is that they're going to cluster in certain areas, representing the different forms of secondary structure. Because remember that it's not like the Ramachandran plot is dictating where these things go. They're plotting the points where these things naturally go, and the things naturally go that way in order to minimize that steric hindrance and maximize the favorable hydrogen bonding ability. Okay. So that is like the normal case, but this is typically when you see a plot like this, you can see like no glycine or pre-proline or proline. Basically what happens is that if you have glycine or if you have proline, these can take on weirder angles. Glycine, because it's small and flexible, and proline, because it's sterically hindered, it's got that backbone that folds up on itself. It doesn't have that opportunity for hydrogen bonding, so it doesn't like to be in those normal secondary structures. They don't like to have it there because they can't hydrogen bond with it. And it can also sometimes take in the cis condition. So you'll see glycine and proline, as well as the kind of the residues that are like right before proline, that are going to be sterically hindered or have different angles. And so you'll see them on different places in the plot. This is another interactive page that is nice for looking at things like this. So it's actually going to let you look at various residues and various stretches of proteins, and you can do this for any protein that you're interested in. And you can go, or you can just and you can go to like all these different angles. But what you'll see is basically you can have that proline. When you have a proline, it's kind of going to be able to adopt weird angles, because amino acids typically take on those characteristic angles. What happens is that when structural biologists solve a structure, they use X-ray crystallography or they use cryo-EM or something like this. They generate experimental data that they then fit a model into. So what we're looking at here is an example of a ribbon diagram representation of such of a model. And the scientists, they actually placed all the atoms in the model to kind of like fit the data, but it's possible that they didn't fit it quite correctly, that some of the atoms maybe aren't in the perfect, in their actual arrangement, that some of those angles that they modeled in might not actually be correct. And one way that they look to see if their model looks okay is by looking at a Ramachandran plot and looking at whether there are outliers. So what we can see is that if we were to go to the protein data bank, which we'll do a lot in class, this is going to allow us to look at all of those structural models. And we can also see kind of like validation things for it. So you can see here that it has some Ramachandran outliers. And basically in red, it's kind of like what it's doing is it's comparing it to other structures and saying, okay, well, this one has more Ramachandran outliers than most structures that were solved that have a similar resolution. So this could indicate that something is off with the modeling a little bit, or it can also indicate that there's kind of cool things going on in the protein, because sometimes you can have outliers that actually have functional significance, not just they're modeled wrong. So let's actually go and look at a Ramachandran plot for this protein. If you look up the protein's PDB code, so this is session code, in this case 1BHL, which is also what I use to look it up in the PDB. If you were to open that in Proteopedia, well, now what you can do is you can actually do a Ramachandran plot of it in its interactive article. If you go over here and you click load full, now what's going to happen is now if I go and I right click, it's going to give me various options. If I press on console and then I type in Ramachandran, what's going to happen is it's going to show me a Ramachandran plot. And now I can see, well, yeah, it looks like we had a lot of those beta strands in that protein. And we're seeing that we have a lot of points over here in that beta strand region. It also had some alpha helices, and so we're going to see alpha helices over here. Now you'll see some points scattered around. We would expect most of these to be glycine. And in fact, here we have a glycine, here we have a glycine, here we have a glycine. So we're seeing that glycine is showing up in all of these different places because glycine is so small and so flexible. But here we have like a glutamate. We might have some other amino acids that are kind of in positions that you wouldn't normally expect to find them in. And this could be why they were then showing as like that we had Ramachandran outliers. In addition to having those kind of like core regions, which are in here often in like a dark color, or they have this darker color where you'll find more dots of them. There's also kind of like these allowed regions, and there's generously allowed regions. And so maybe those glutamates were over in here. But basically, if you see things outside of those regions, then that would be considered a Ramachandran outlier, and that would be something that you might want to go and look into. Okay, so if you look over here, you can actually, going back to that original Ramachandran plot tutorial post, you can actually go and you can look at structures in their Ramachandran plots within this page itself. So here we're seeing a structure with a lot of alpha helices. And if we view its Ramachandran plot, as we would expect, we're going to see a lot of points that are going to cluster in this, in the alpha helix range. And then we have some outliers, which are going to be things like typically our glycine. Okay, well, let's view another one. Let's view this concavalin A. Now, if you click it, it's going to take you to the actual page for it. And so here's the page for it. We see that it has a lot of beta strands. And so you should be able to identify beta strands, know if they're parallel, those sheets are parallel or anti-parallel, that sort of thing. I don't need you to know where things cluster on a Ramachandran plot for a beta strand or for a beta sheet, but I do want you to know that there is a place where they cluster, and they cluster in this place because those are the angles that are favored for a beta strand. And so basically, these angles are favored because those are positions that are allowed based on steric strain or steric hindrance. And they're also places where at those angles, the amino acids are able to organize their hydrogen bonding groups to have ideal hydrogen bonding with the other strands. And so that's why you see these characteristic angles taken. What about one final one? Let's look at acetylcholinesterase. So here we're going to see that we have a structure that has a lot of alpha helices and beta strands. And so if we look at a Ramachandran plot, as we would expect, what we're going to see is we're going to see a lot of points that are in the alpha helix range, and a lot of points that are in the beta strand range, as well as some other points that are in the regions where you'll find things like left-handed helices and things like this. You'll also see a lot of glycine just like floating around in weird places, because remember, glycine's small, flexible, it can take on those turns. It's really great for all sorts of stuff. But you'll also see a couple of points that, or at least one point that's kind of weird. And so as you go and it points out the serine 200. And so basically, if you go over here, you'll see that this is serine 200. So normally you wouldn't find a serine over here. It's outside of the preferred regions. But basically, as they say, this is part of the catalytic site, and it's actually a really important residue for this protein. And so sometimes you'll see that the really important things are kind of in an awkward angle. And so these unique values of phi and psi are going to help position it for the catalytic role that it needs to play. So what I hope that you take away from Ramachandran plots is not where things are, like not what exact angles are, not even what the phi and the psi angles are, like which one's which. Just know that there's these places, only these certain places on the peptide backbone where rotation is allowed. There's only rotation at these places because in between them, when you have a peptide bond, that's going to have this resonance. It's going to have this partial double bond character. It has to be confined to a plane so that those p orbitals can line up. If you were to try to twist around it, you'd be breaking that resonance. You'd be making these things super duper unhappy. And basically, for most of the amino acids, it's going to be like super clashy if you were to go to cis form, but proline, it's clashy either way, and so it can go to the cis form. But this is going to require the help of an enzyme, because when proline is put into the protein, basically the ribosome is always going to put things in the trans form. And then if you want to actually rotate around that bond to get a cis protein, that's actually going to take the help of an enzyme. So bottom line is you're going to have these restricted planes, and when you link them up, you're going to get a lot of these planes linked up, and there's going to be certain places where you're able to rotate on either side of the alpha carbon. You can rotate freely there, but, well, not truly freely, because you're going to have to deal with steric hindrance. So as you rotate around those bonds, you're going to have clashes, and especially if you have big bulky things that can further restrict like which angles you're allowed to be in. And then we end up with places where you're most likely to find various amino acids. And if you take into account wanting to align your hydrogen bond groups for optimal hydrogen bonding opportunities with your backbone, well, then things are going to be confined even further. And so there are specific angles that are preferred that you'll commonly find in various secondary structures, things like sheets, things like alpha helices, things like turns, all these various places. And so you'll see that you'll find them clustered on different parts of the Ramachandran plot, because this Ramachandran plot, well, it's plotting the angles that are found. And because these angles are constricted, and then they're further refined based on those hydrogen bonding opportunities, they're going to cluster in specific places. And therefore, you can kind of identify these allowed regions and the mediumly allowed regions and the generously allowed regions. And when structural biologists look to see if their models are correct, they'll often look to see if they have their residues are angles, their phi and psi angles are clustering in the right locations. And also, though, there's often exceptions are made for glycine and proline, because glycine, it's going to only have a hydrogen, it's going to be small and flexible, it's going to be found in all sorts of regions where you wouldn't find other ones. With proline, well, here it has this weird sterically hindered backbone, and so it's going to also sterically hinder the thing before it. So often you'll see like Ramachandran plots will show regions and they'll show points excluding proline, glycine, and the things before proline, because it's going to have to deal with this with proline's bulk.